Hello, this is Ed Chapman, and this video cast is going to focus on the work of Gregor Mendel, often referred to as the father of modern genetics. Mendel was the first person to come up with the idea of what we now know as a gene. And as you remember, genes are loci, or places on chromosomes where alleles are located. And alleles are different forms or different potential forms of a gene. Mendel figured out that inheritance is actually controlled by a pair of alleles. He called them factors, but we now know that they're genes. All right, you get one from your mother and one from your father. And today, we identified that Mendel's factors are actually genes. Different genetic codes produce different alleles, which are just different versions of the same gene. For example, different versions of genes for eye color or hair color, or dwarfism versus normal height, things like that. Mendel's work was quantitative. People have always known that children look like their parents, and people kind of assume there was a blending action going on with inheritance kind of acting like mixing paint. Okay, but Mendel was the first person to figure out that genes are actually particular, are particles. They don't generally blend, at least in the traits that he studied. And he studied pea plants, and he cross-pollinated them, or he let them self-pollinate, and he got many, many generations of data. And he identified that there are things called true breeding plants and what he called hybrid plants. Mendel's most famous cross was his first one. It's called his F1 cross, and this is where he crossed two pure breeding but different traded plants. In this case, we're crossing a purebred purple with a purebred white, and he noticed in the F1 generation, the first generation of offspring, he got all purples. Okay, so from this, he figured out that purple was what we call a dominant trait. Now, when he crossed or self-pollinated the F1 generation, he got a mix of purple plants and white plants, okay, or purple flowers and white flowers. And he always got the same ratio, 3:1, which is the same thing as 75% to 25% for the F2 generation, or the second generation of offspring. Okay, and from this he figured out something that we now know as Mendel's law of segregation. And the law of segregation says that when an organism produces haploid gametes, the alleles pairs have to break up. They have to separate. Okay, so when gametes are formed, each gamete gets only one allele. It can never get both. Okay, we now know that happens because of the separation of homologous pairs during meiosis I. Now, because 100% of the F1 generation turned out purple, Mendel was able to say that purple is what we call a dominant trait and white is what we call a recessive trait. So that gives us Mendel's law of dominance, which says that in hybrids, excuse me, the recessive trait is hidden by the dominant trait, which shows. Okay, some useful genetic vocabulary to know are the words homozygous, heterozygous, phenotype, and genotype. Okay, I'm pretty confident you've heard these words before. That seems to be the report I get from juniors and seniors that saw this in freshman biology. Okay, you also need to know what a test cross is. Let me show you what we're talking about here. If you have a purple flowering pea plant, for example, and you just know it's purple, okay, you don't know whether it is homozygous or heterozygous for the purple allele. So what you do is you cross it with a known genotype, which of course is white, because if a pea plant is white, it has to be homozygous recessive. And you're going to get one of two scenarios. You're either going to get all purple offspring, okay, or you're going to get a mixture of purple and white offspring. If you get all purple offspring from the test cross, you conclude that the purple parent must be a purebred. If you never get white offspring, if you get a mix of purple and white, you can conclude that the purple parent must have been a hybrid genotype. All right, now monohybrid crosses and dihybrid crosses are similar. They just differ in the number of traits that you're crossing at the same time. Monohybrid crosses, just like they say, is a cross of one trait at a time, whereas dihybrid crossing is when you cross two traits at the same time. And Mendel did a whole bunch of dihybrid crosses, and in this diagram he's crossing two traits, which is pea color and pea texture. Okay, so a pea can be yellow, it can be round, or it can be green, or it can be wrinkled. Okay, so yellow and round are the dominant traits, and green and wrinkled are the recessive traits. And when Mendel did dihybrid crosses, he always got the same ratio. He got a 9 to 3 to 3 to 1 ratio. And from this, okay, he figured out his third law, okay, the law of independent assortment, which means that each trait is assorting or separating. The alleles are separating with no respect to the other alleles. Okay, each pair of alleles is independent of all the other pairs of alleles. Okay, now Gregor Mendel's work and the genetics that he observed in pea plants is governed by the laws of probability. Okay, and we can use the laws of probability to figure out genotypes of offspring without, or probabilities of offspring without actually doing Punnett squares. Okay, so let's say you have this dihybrid cross and you want to figure out the probability of getting big A something, big B something. All right, how would you do that? Well, hold on. I didn't turn on my little pen here. Now I got my pen. Sorry about this. Play slideshow. All right. Okay, so now I can get to my little pen. All right, I want to write red. All right, so what's the probability of getting big A something with big B something from this cross? Well, what you do is you look at just the A's. So if you look at the big A little a crossed with big A little a, this is a very simple Punnett square. All right, so what's the chances of getting big A something from this cross? It's going to be 3/4. Okay, because if you do this Punnett square, okay, you cross big A with big A little a, you get this one, and this one are going to have it one big A or two big A's. So that's 3 out of 4. And the same thing for B's. The B's are going to follow the same pattern. Okay, I'm not going to draw it again, but it's the exact same proportion, 3 out of 4. So what's the probability of getting this? It's going to be 3 times 3 is 9. 4 times 4 is 16 because you multiply these together. So it's 9 out of 16. So that's how Mendel got the 9 in his 9 to 3 to 3 to 1 ratio. All right? So that's a way that you can calculate without having to do the Punnett square. So there's several of these. I'm not going to take the time to do it because I'm running short here. Now, there are traits that don't follow Mendel's law of dominance. Okay, Mendel worked out these 3 laws, but not all traits follow them. Some people fault him for focusing only on the complete dominance that he saw in pea plant traits, but there are other things. Okay. So here's a summary of Mendel's 3 laws. But there are other things about genetics that do not follow Mendel's law of complete dominance. We have incomplete dominance, multiple alleles, polygenic inheritance, and sometimes the environment can have an effect on phenotype that's separate from a genetic effect on phenotype. Okay, incomplete dominance is also known as blending. That's when 2 phenotypes combine to make an intermediate or middle third phenotype. The example that your book uses is red snapdragons crossing with white snapdragons to produce pink snapdragons in the F1 generation. All right, so in Mendel's pea plants, remember the flower color was completely dominant. Purple was completely dominant to white. But in snapdragons, as you can see here, red and white cross to make something in between, kind of a pink color. Okay, multiple alleles is when you have more than 2 alleles possible at a certain location. Another name for multiple alleles is codominance. Okay, for this class, we're going to kind of make them the same thing. Okay, so codominance is pretty much what we see in something that produces the blood types that humans have. It's called the ABO blood typing system. All right, so in the ABO blood typing system, I don't know why that's cut off up here, but here you have 2 dominants are possible, a big I with an A superscript or a big I with a B superscript. There's 2 possible dominants, but only 1 possible recessive. So you have a total of 3 alleles possible at this locus. Now, everybody only gets 2, so if you get 2 of the dominant A's or just one of the dominant A's, you have the phenotype type A. And the same thing for type B. AB is a complete hybrid for this, hybrid dominant. And if you're purebred recessive, you get 2 of the little i's, you have type O. All right? So what makes this different from Mendel's genetics is you don't just have a big A and a little a. Here we have 2 dominants and a single recessive for a total of 3. Okay, polygenic inheritance is actually what's very common in human phenotypes. For example, skin tone. This is when many genes are controlling a particular trait. The best example I can think of is skin tone. So, for example, at least 3 different loci are contributing to the tone of a human's skin. So that means if people with different skin tones produce offspring, they can get a whole range of skin tones that are very unpredictable for the offspring. All right, you can kind of see from this Punnett square here how complicated it can get. Okay. Notice that there are many different genotypes that produce the same skin tone. All right, what that means, if you look at this chart, this right here is kind of a medium skin tone. This right here is very similar to it, but they're actually produced by different genotypes, even though they have the same phenotype. So it gets very complicated. Okay, there are environmental factors that control how something looks that has nothing to do with the genes. Okay, this is the nurture part of the nature versus nurture debate some of you may have heard about. The best example I can think of this is flower color in a plant called hydrangea. Okay, here's a picture of some hydrangea flowers, and you may look at this and say, wow, the blue one and the red one, they must have different genes, but no, they don't. They're genetically identical. But what causes the different flower color is an environmental condition. It's the pH of the soil. So if you grow hydrangeas in acidic soil, you get blue flowers. If you grow the exact same plant in basic soils, you get pink or red flowers. And if your soil is neutral, you kind of get this muddy brownish mix of the two colors that nobody likes. Okay, another example of environment affecting the form of an organism. Here is a spruce tree. Here it's normal. All right, over here, this is what the tree looks like growing in normal conditions. But if it's growing on like a mountaintop that gets wind from one direction all the time, the wind dries out the buds on this side, so you end up with this flag type tree, they call it. And if it's growing in a very, very cold, very windy environment, sometimes the tree is actually flattened out along the ground. And this is what you get with freezing wind, like in mountaintops. So the point is, a spruce tree may look completely different depending on what environment it's growing in. All right, thanks for listening.